Hello everyone, I'm Dr. Arpita here. The topic for today's discussion is numerical problems on Hardy-Weinberg equilibrium. Taking into consideration that you are aware of the theoretical part of Hardy-Weinberg equilibrium, I'm straight away moving into the numericals. Okay. See here. This is the picture of Godfrey Harold Hardy and Wilhelm Weinberg. They have given us a wonderful concept to study population genetics. Fine. A fundamental principle in population genetics stating that the genotypic frequencies or genotype frequencies and gene frequencies of a large randomly mating population remain constant provided immigration, mutation, and selection do not take place. So this concept, its explanation, conditions, everything has to be ready at your fingertips. You must know everything very well. So as I told you, I'm taking into consideration that you know the theoretical part of it. I'm not discussing all those things here in this lecture. I'm straight away moving on to the numericals. Okay. So see, as we all know, in a population at Hardy-Weinberg equilibrium, the allele frequencies remain same from generation to generation. Now see, please note these down. This will help you when you're preparing for your exam. So what did I say? In a population at Hardy-Weinberg equilibrium, please don't write this type of abbreviations in your exam. This is just for explanation purpose. Please do write Hardy-Weinberg nicely. Hardy-Weinberg equilibrium, what happens? See, allele frequencies remain the same from generation to generation. This is what you already know. Remain the same from generation to generation, and genotype frequencies and genotype frequencies remain in the proportion p square plus 2pq plus q square, right? Now, our entire discussion, concept, whatever you may call it, will pivot around this particular formula, correct? So, see, if I just write it down, it will be better for you to understand. See, for F1 and F2 generations, mind it, huh? Genotypic frequency of capital A is p squared, right? The dominant counterpart. Then genotypic frequency of capital A, small a. This is the dominant homozygous counterpart, right? This is heterozygous condition. This is 2pq, right? Then genotypic frequencies of the recessive part, homozygous recessive. What is that? Q square. Now, if I ask you then what is the allelic frequency of capital A? allelic frequency of capital A, what would you say? It will be P square plus half of 2PQ, which is equal to what? Yes or no? Yes or no? See, P in the denominator and P here gets cancelled out. Correct? Yes or no? Then what do you get? What are you getting? P square plus PQ. Here, if you open this bracket, you are going to get that only. I've just taken P common and have put P plus Q in the bracket. And this is equal to what? This is just equal to P. Why? Because we know according to Hardy-Weinberg equilibrium, P plus Q is equal to 1. If you replace this, if you substitute the value of P plus Q here, you are going to get P. Right? So allelic frequency of capital A becomes P. Same thing if we do for allelic frequency of small a, what is that? Q square plus half of 2PQ, right? Which is also equal to Q since this is the condition. So allelic frequency of capital A is equal to P, allelic frequency of small a is equal to Q. Clear? This part till here it is clear? Okay. Now see if we come to gene frequencies. Correct. If we come to gene frequency of say the dominant counterpart only. Suppose we take it as M. Just make a note. These are actually very easy if you understand the concept. And once you understand the concept, there is no way anybody can deduct a single mark from your paper. So see, then what will be the formula? See, gene frequency of, what did I write here? Capital M. You can take any alphabet. What will it be? Total number of M alleles. Okay, total number of M alleles divided by total number of alleles of all individuals. Clear? This is the gene frequency of capital M. Now, if I say genotypic frequency of MM, note this. Genotypic frequency of capital M. What will it be? It will be total number of MM individuals by total number of individuals in the population. Total number of individuals in the population. Not this, population. Right? Now you will have to keep these two equations in your head. You will just not forget this. If you are planning to forget this, that means you are purposely inviting trouble for yourself. So these two things you are just not going to forget. Is that clear? Okay, now, based on these little things what we have just discussed, we are going to solve few problems. Let's see what have we learned and how much can we achieve by learning these little formulas. Correct? Fine. Now let's talk about a problem. Okay, I'm writing this. See, I've not come here to give you an opportunity to copy what I'm writing. Please use your gray matters as well. Try to solve these problems with me only. Please don't wait and see whatever ma'am is writing. Let's copy. Don't fool yourselves, okay? So let's see the question. What is the question telling you? The following genotypes are observed in a population. The following genotypes are observed in a population. What are the genotypes they are observing in a population? It's given. See, what's that? Capital H is equal to 90. Capital H, small h is equal to 60. Small h is equal to 50. Okay. Now see, two questions are there. You are supposed or you are required to find out what allele frequency. This is one question. Second question is what? Number of individuals in the population. Correct? These are the two questions. The two questions are very much different. First of all, we are going to look for what? From the given data only we can find a lot. Yes or no? First thing what we can see is total number of individuals. Total number of individuals. How many? 200. From where are we getting 200? See, if you add this, you get 200. Okay. So your total number of individual becomes 200. Now what are we going to do? We will first calculate the genotypic frequency. Correct? Genotypic frequency. I'm going slowly, step by step, so that every bit of it gets settled in your head. This should be a one-way process, unidirectional process. It should not go in and then it should come out. No, it should go in and it should settle there permanently. Okay? Okay. Genotypic frequency. Please calculate before I do. Don't just copy it blindly. Simple mathematical calculation. It is nothing so great here. Yes or no? Very simple. Then this one. See, I am only calculating from what's already given in the problem. I'm not doing anything exceptional here. Yes? Yes or no? So these are the three results that you have got. This one, and this one. Correct? Correct? Okay. Now, therefore, your allele frequency of capital H in the total population, your allele frequency of capital H in the total population. What is that? This one we are going to calculate. This one will be this plus half of this, which is equal to 0.60. Yes, agree? So your first question is answered. Allele frequency of capital H. Here I should have written allele frequency of which one? This. And this. So your answer is this. Allele frequency of capital H in the total population is, excuse me, 0.60. See here, genotypic frequencies of both capital H and capital H small h are considered. This one, this capital H as well as this capital H small h, everything is considered here. Fine. Okay. Now, story doesn't end here. This is one part. What is the next part? Therefore, allele frequency of small h is equal to what? 0.25 plus half 0.30. Agree? Which is equal to 0.40. Right? This is the allele frequency of the recessive one. Now, this formula is something which we know. P square plus 2PQ plus Q square. This is what? P plus Q whole square. This is something we know. Now, if we put the values in the equation, what are you going to get? 0.6 square plus 2 into 0.6 into 0.4 plus 0.4 square. This is what you are getting. This means what? 0.36 plus 0.48 plus 0.16. Yes or no? Therefore, percentage of HH individual is, therefore, percentage of individual is what? 0.36 into 100. Yes or no? That is 36%. Clear? Now, what was the question? Number of HH individual. No one had asked you about the percentage. Here lies the trick. Number of HH individual, that is capital H or dominant, whatever you may say, in the population is what? 36% of 200, which is 72. So your answers are what? One is this one. This is one answer. The other answer is one. Is it easy or not? Yes, good. This is really easy. So you have got an idea, right? How to solve it. Yes or no? You have got an idea, right? Great. Now, we will talk about another question very similar to this. Okay. So our new problem in hand. What is this? See, the following blood types are obtained from a sample of 205 individuals or 205 Anglo-Indians. See, if you are seeing this name, don't get confused. Nothing is there. Try to understand the message in the question. That means you have a total of 205 individuals. Okay? Right? Actually, they have done this addition for you. That's all. You can also find it out. That's not a big deal. Now, the question is what? See, calculate the gene frequencies of M and N. Capital M, capital N. So, how to start? So, you have total number of individuals, right? Which is 205, 205. Correct? Since it is a diploid population, we can easily say total number of alleles, total number of alleles will be what? 2 into 205 since diploid. Had it been triploid, we would have multiplied it with 10. 410. Since each individuals have 2 alleles, we are multiplying this with 2. Now see. Now see. M alleles will be equal to what? 123 into 2, which is 246. Yes or no? Wherever you have a doubt, please stop me. Mn alleles 72 into 2, which is 144. N alleles 10 into 2. Correct? 20. Is that clear? Therefore, total number of M allele is what? 246 plus half of 144. Yes or no? From here we are getting, this is a summation, this is a summation of, not summation, this number is formed with association of two alleles, M and M. So why will we consider it as a whole? We will only take M. The concept should be clear. This is very easy. So this is equal to 246 plus 72. Yes or no? Which is equal to 318. So total number of M allele becomes 318. Same way if we do total number of N alleles will be what? 20 plus half of 144, which is equal to 20 plus how much? 72 is equal to 92. This is also one part. Now we will consider the question gene frequency. How will we calculate that? Gene frequency of M. Gene frequency of capital M. Yes or no? This many out of the total. So what is that? 0.776. Clear? And this is one answer. Then gene frequency of N is how much? 92 by same total number of individual, which is equal to 0.224. So these are your two options, your two correct answer. Fine. So there will be many other problems. I'll try to make another lecture, or I'll come to you in another class with a few more problems. In the meantime, please stay tuned. Practice different problems from different types of books. You will get a lot of exposure. You will get different types of problems, and the more you solve, the better it becomes. I mean, the better the concept becomes. Okay, the clearer the concept gets. So please don't uh, don't get afraid of doing all these things. There is nothing to get afraid. It is really very easy. It's just that you need to understand the concept. That's all. Okay. So thank you so much for attending the lecture. If you have any doubt, any confusion, please feel free to get back to me. Okay. Till then, bye-bye.